lots of people filing into the room, and go ahead and kick us off. So happy Friday, everyone, and welcome back. Thank you for taking the time to join us for this upcoming Patient Perspectives and Fireside Chat and Day 2 of the Rare Drug Development Symposium, hosted by Global Genes and the Orphan Disease Center at the University of Pennsylvania. My name is Susan Brizendine, and I'm head of engagement programs at Global Genes, and I'm really pleased to have the honor to kick you off today. So yesterday we heard lots of interesting discussions, exciting discussions, and the Global Genes team had just a few takeaways and things that struck us in particular that we wanted to share with you before we got started today. So I'm just going to share some kind of observations, a few insights, and some highlights for what you can expect today. So first of all, some news though. I'm not sure if many of you saw this, but in FDA Voice this morning, the FDA had an article about their commitment to minimizing the impact of COVID-19 on progress in care and clinical trials in rare diseases. So that was really good to see. And we just wanted to encourage you to make your voice heard, because they have a public comment period in something they're working on through July. So we would just, again, encourage you to make sure you have a voice and contribute there. So a few observations from the Global Genes team, and I think everyone who was part of yesterday's meeting. Chris Austin, I mean, just wow. I think we were all amazed by his passion and his boldness and his, you know, frankly, his intolerance to a certain degree. It was so encouraging and somewhat contagious, right? I think everybody was kind of on fire by the time he was done speaking, and the comments were just flooding in about how much people enjoyed his talk. So thank you for that great feedback, and thank you again to Chris Austin. So much interaction during yesterday's sessions, great dialogue, and you know, I don't know about you, but I didn't want that hour to end. It just didn't seem like enough time. And we heard about some virtual hugs that were happening in some of those Zoom rooms. So that is just fantastic. And then we were blown away by the Cure Accelerator finalists, such exciting and interesting research going on, and we're all on the edges of our seats to find out who's the winner. I know that was a difficult vote for many of us. And that takes place right after this session. So a few insights. Jim Wilson said it, Chris Austin said it, we need you, right? That was the message to patients, that patient membership on research teams is disruptive technology. It's so critically important. And we've come such a long way, right? I mean, historically, rare was rare and mainstream didn't care, but now we've made so much progress as a patient community, as a larger rare disease community, in patients driving research forward, right? It's been a huge paradigm shift, and patients are bringing so much value to these partnerships with industry and with researchers and academia. I mean, they're showing up to these conversations with their own data, with genomic sequencing data, with clinical trial data, with natural history studies and national, you know, registries and their electronic health records. It's just so exciting to see that collaboration that's being driven by the patients. And we talked about that yesterday. We'll talk about it more today. And there's challenges ahead, right? But we're all in it together. That's why we're here today. We've got industry and patients and academia and researchers all together to get over what I know the rare disease community just calls them speed bumps, right? They're just something we need to get through with careful consideration, but there's drive and eagerness and such an investment. So a couple other things, insights from yesterday. Chris Austin addressed it. We saw in other places that the pace is painfully slow with development, and that needs to change. We've got to figure out to improve our efficiency, and COVID-19 was, you know, something that set a new standard for collaboration and openness and efficiency that we hope will be applied to the rare disease community and will really stick. The diversity and inclusion roundtable yesterday was very powerful. We talked about the importance of transformation first, that we have to change our mindset and then performance will follow, that listening and learning and including and asking and involving is imperative, and one size doesn't fit all. And that patient impact is what counts with regulators, right? We have to start from the end in mind with the impact and then back up into the research and strategy. So today, their packed day of rich content, opportunities to learn and connect, and just some topical highlights. First of all, overcoming obstacles in doing research in small populations. That's first up today. There's a government funding workshop. We're going to learn more about artificial intelligence and working together between academia and research. Today you get to pick a different roundtable to join, or if you loved the last one, go ahead and join that one again too, if you still have some conversations to be had. And then at the end of the day, we have a fantastic panel to send us on our way, and it's being led by Global Gene's very own Christian Rubio, who is the head of our strategic advancement team. So I also have to thank our sponsors, because as you well know, we could not do such extensive programming yesterday and today without the help of these supporters. We are so grateful, and I know many of them are in the room, so we just wanted, on behalf of the entire rare disease community and the Orphan Disease Center and Global Genes, thank you. Thank you so much for your support. So I know we're eager to get to our speakers. I have just a few housekeeping things to get through. First of all, attendees will be muted by the moderator to reduce any interruptions. But we encourage you to use the chat function in the Zoom window for any questions you have throughout the sessions. And the moderator will then present those to the speakers, present your questions. And to open the chat, you just click on the chat button that's located on the meeting controls of your screen. It's near the mute icon. And I think we all know where the mute icon is, because for some reason it's always stuck. We're always stuck on mute. Anyway, if there are any questions that are not able to be addressed by the speakers, if we run short on time, we'll be sure to try and circle back with you and get your questions answered. If you have any technical questions or difficulties with the agenda, just go to our help desk through the lobby, and our Global Genes team and tech team will be happy to help you. And finally, we encourage active listeners. So don't miss this. I mean, I am guiltier than most in multitasking. I don't feel like I'm productive unless I have 3 things going on at one time. I really encourage you to take full advantage of this time and be present. You're getting critical questions asked and answered about rare disease research, so we hope that you'll fully participate. So now I'm really pleased to introduce and welcome our speakers. Nasha Fitter of FoxG1 Research Foundation. She is the co-founder and CEO. And David Rich, who's with Ipsen, and he's the Vice President of Global Development, Rare Diseases. So they will each share a little more background as we delve into the conversation. But right now, I'd just like to welcome Nasha to kick us off. So hi, Nasha.
Okay, thank you, Susan, so much for that great introduction and kickoff to the day. I love Global Genes. The conference was the first thing I attended when I got the diagnosis for Amara a few years ago, and it really set me on my path. So my name is Nasha, and I have a 4-year-old daughter named Amara, and when she was 7 months old, she started having seizures, and shortly after was diagnosed with a mutation in her FoxG1 gene. The condition is called FoxG1 syndrome. And like so many of the parents on this call, that completely changed my life into focusing on what can I do to get a therapy to help my daughter. And what it's turned into now is the larger community. When Amara was diagnosed, there was really not that much known about FoxG1, and I know many of you who've gotten your children diagnosed experienced the same thing. There were sort of pockets of basic science research, but no concerted effort. And so what I'll be talking about with David is what can we do as a patient population to understand our disease better, and that really segments into 2 ways. One is to understand the clinical symptoms, and that's where foundations can really help biopharma, and it helps us as well. So we'll be going into patient registries and natural history studies. And then 2, on the biological side and translational side, what can we do as a patient community to speed that and de-risk the investment for biopharma so we get our diseases picked up. On to you, David.
Okay, well, thanks, Nasha. My name's David Rich, and as Susan said, I work in drug development for Ipsen, and we're focused on rare and ultra-rare diseases. And Nasha and I today are going to be talking about issues such as natural history studies, to learn more about the natural history and progression and patient experience of rare and very rare diseases, as well as ways in which we can partner together for clinical research to generate the data that's needed to bring potential drugs to patients. So we're very much looking forward to that discussion.
Oh, Susan, I think you're on mute.
I told you, everybody's always stuck there.
Yeah.
Okay, fantastic. So let's, you mentioned natural history studies, Nasha. Let's start there. So help, both of you, help us understand why natural history studies are so important. Nasha, do you want to go first?
Yeah, and David, you'll chime in on the biopharma side. Obviously, why is it so critical for patient advocacy groups? There's two reasons for us why it's important. One is we need to ourselves understand our disease better, so we can make better day-to-day decisions. As we're waiting for therapies, we need to make day-to-day decisions for our children. What medications should we give? What are we going to experience? That giving the control to us to really understand our disease. And two is, I have a great mentor, a CEO of a biotech company, who told me early on that if you do nothing else, just get a natural history study done. That will attract biopharma to your disease. And in terms of natural history studies and patient registries, the few learnings I have to offer is, one, you have to make sure that you as a patient advocacy group have full control over it. So if there's a biopharma that wants to conduct your natural history study, that is not going to serve you in the long run. That might serve you in the short run, but not the long run. You want full control of that, so then you can have multiple biopharma companies that can access that data. In any disease, there's a lot of patient burnout. Once a patient enrolls in one study, there's slim to none chance of them enrolling in a second one. In our rare communities, that's a real threat, right? We have so few patients to begin with. So it's really important that you choose platforms that are patient controlled, advocacy group controlled, and multiple parties can access the data. With academic centers, this becomes a big problem, right? Academic centers don't like to share data with other academic centers. There's a lot of rules and regulations. You have to be really careful when you look at contracts. So my recommendation is choose open platform when you do this. The second part that I just wanted to explain is that there's two parts of data. One is patient-reported outcomes, which are important, but it's also important to note that the FDA, there's very few patient-reported outcomes that they will take seriously and take inside for a submission. So that's something to think about. But it's still very important to get patient-reported outcomes. And the second part is collecting medical records and getting the clinician-reported outcomes, which are data points that the FDA will validate and take forward. And so it's really important to figure out platforms where you can do both. And it could be two separate platforms, but this is how you want to think about it: patient-reported and clinician-reported outcomes. And then when you get those medical records, who is going to be abstracting the data from those records, normalizing the data, creating a data set. These are all things that, we can go through later in the presentation. And then the third thing is getting a strong PI. And I cannot stress how important a strong PI is. This is a geneticist, a genetic counselor. And my recommendation is find someone who's young and hungry, right? You don't have to get like a famous geneticist that everyone is trying to get a call with. You just need to get someone who's good, who wants to make a name for themselves and is really going to invest in your disease. That's what we did at FoxG1. Actually, my daughter's genetic counselor at Stanford, young, hungry, we've taken her on, and she has been such an integral part of all of our research and has run our patient registry, is now running our natural history study. So that's really, that's very important. And I'll stop there because I want David to get a chance to let us know on the biopharma side, why are these studies so important, and what can we do?
No, thanks, Nasha, and couldn't agree more. And so the example I'm going to use for us is we're very interested in developing therapies for a rare disease called FOP. Which is a very rare bone disease. And so actually the first thing that we did was start a natural history study because there wasn't one. And so that's a very good way of finding out, event rates. So for example, for FOXG1, it might be numbers of seizures in a year. For us, it's the volume of extra bone that's grown. So without a natural history study, you don't know those types of aspects that are centrally important to these rare and very rare diseases. And why that's important is because then if you want to start a drug study, you need to know what you're going to be looking for, what you're going to be measuring and counting. And actually there are a natural history as we started, and say we started that way before any of a study involving a drug, because then you can start building your network of the patient advocacy groups, like Nasha has said, as well as the investigators and those very specialist centres around the world that are treating these very rare diseases. There are also other benefits. For example, in speaking with your regulatory agencies, depending on your rare disease, or sometimes a natural history study could be used as comparative data to a drug study. And that could, in some circumstances, mean that you don't need to have a placebo control, for example. And so we've been able to do that, in one of our cases. So, but what was good is the benefit of starting a natural history study first was that as soon as we started a study with an experimental drug therapy, patients were able to transfer from the natural history into the study where there was a potential treatment on offer. Of course, at that point, we didn't know whether there was a benefit because it was experimental, but it was a very nice sequence of events. So natural history study first, learning a lot about the disease, then we can design and build a study with an experimental treatment, and subjects could come in and receive that experimental treatment as part of the study because we already had relationships with the advocacy group as well as lots of the clinical trial sites around the world. So really, really useful.
Yeah, so based upon your responses, what I'm hearing is natural history studies critically important for both parties. Advocacy shouldn't wait for industry to do that, to initiate that kind of study. They should go ahead and get that started. And in some cases, it's potentially easy transition to some clinical trials if you've got some people enrolled in that natural history study. Okay, very helpful. So around selecting endpoints in a clinical trial, David, can you tell us what are some important areas to consider and think about?
Wow, I mean, we could spend the rest of your morning talking about that. So of course, you need to be able to measure something that is in itself measurable. So something springs to mind. I mentioned seizure rate for something like FOXG1, or for us, volume of bone. In themselves, they are quite easy to measure. And so what a natural history study could inform you is what other endpoints, both patient reported, are also important in the disease, because once we've got data from a clinical trial in our hands, of course, we have to go and speak to regulators like the FDA in the US or the EMA in Europe. And they're interested in endpoints, but there are a lot of endpoints in rare and very rare diseases that aren't validated. So a regulator might say, yeah, that's really interesting that you've managed to decrease this by this much, but what does it mean for patients? So we have to be able to then prove that it's a clinical benefit to the patient. So that's why having patient involvement and advocacy group involvement in designing what endpoints you collect is really important, because of course what we need to do is not just report on the endpoints and showing some significant benefits in respect of those, but that makes a positive difference in terms of a clinical benefit to the patients as well. And then after that, nowadays, we have to have a body of evidence as well for payers. And they're often interested in different things. So at the start of a study, you will need endpoints that you can measure easily to show that you've had a positive effect on a disease. Then you need to also collect endpoints that can help you demonstrate there's been a clinical benefit to the patient, and then collect the endpoints that will be interesting to payers around the world so that patients can get reimbursement.
Yeah, and sorry, Susan, I was just going to say
just real quick, Nasha. And isn't it sometimes the conclusion that what's interesting in terms of industry perspectives or even regulator perspectives on the impact of the patient, they might have different points of view on that, right? About what is a meaningful benefit.
Oh, it's very rare. It's very rare that they are together.
Yeah.
So you have to think about all that in advance, because once you've started your study, it's too late.
Right.
Yeah, and actually, you know, what's interesting, we asked a survey in our patient registry, if you were going to get, you know, put your child in a clinical trial, what are the most important things that you'd want? Why would you do it? And we thought seizures would be number one because they're devastating, but actually it wasn't. Seizures were last on the list. Communication was number one. And that's what patients are most interested in solving. And I'll say that on the patient registry, I think one thing to be careful of from an advocacy group perspective is you don't need to ask every question. A lot of the information is in the medical record, and you just want to be careful because you want patients to keep coming back to the platform, to keep submitting survey questions. You want it consistent. And if your surveys are extremely long with a lot of unnecessary questions anyway, you're just going to have burnout. So it's really, you have to be smart about what are the areas that are not in the medical record. So for example, sleep, right? A sleep survey is really beneficial, and that's data that's not in the medical record. So just to be smart about what are the questions you're really asking patients to fulfill. You don't need to ask everything.
Mm-hmm. Great. And so in terms of developing a research plan, what would you say are the most important aspects to entice both academic researchers and biopharma to work with you, a patient organization?
So the way we really focused on this from day one and put a strategy in place on how can we de-risk the investment. And there was basically three areas that we focused on. One is we want to show that symptoms can be reversed and at different time points in a patient's life. Two is we want to show what are the biological characteristics of this disease, that we understand the pathways really well. And then three is that we have some proof of concepts for translational work. And so that's how we started our strategy. And if we look at, and you know, this is advice that I wish I had had day one, when you look at symptom reversal, having a conditional rescue mouse model is so critical. And the thing I will say is that there's multiple ways to develop this conditional rescue model. And my honest advice is it's expensive, these mouse models, but go and invest in a couple of different mouse models for conditional rescue. There's different ways to develop it. And what we went through is we waited a year for our first model and then it didn't work. FOXG1 wasn't expressed. We had to go do another model. And, you know, if we just invested in 3 models, we would be in a different place. And most biopharma, they won't even talk to you unless you can show that there's symptoms that can be reversed. So really invest in that rescue model. Research labs that are good at this, that have done it before. It's just such a critical model. Second is to look at patient-specific models. And to do this, you know, to look at, and this is where your patient registry becomes really critical. The most important thing you can ask from a patient registry is having patients submit their genetic testing report so you can validate they even have the mutation that they say they do. And 2, so you can actually start putting like a list of genotypes together and you can start to understand, okay, what are the different mutation variations of my particular disease? And then you pick a few different mutations that can cover the gamut, and you can create then mouse models and IPS lines that represent the gamut. So you pick a few. And for both of these, you want to be really careful on the labs you pick and then characterize these models. And that's how you get to the point where you understand the biology and at a cellular level, your diseases. And it's really great to have 2 asset bases, right? And this is what biopharma wants to see, that you have characterized cell lines that they could test on. And you also have some mouse models that they can test on. And the last is to think about high throughput screening. Small molecule drugs, ASOs, there's a lot of different things to look at. And we looked at zebrafish as a great model to do high throughput screening. There's worms, there's different types of models that you could try thousands of drugs quickly. And so you want to think about that. And the last thing I'll just say, we also learned this the hard way, and this goes into understanding your disease and thinking about what are the translational opportunities. So antisense are a whole new category that have emerged. But you have to think about what are the models that are required to test an antisense therapy. In our case, we need a humanized model. Again, I wish I knew that a couple of years ago. We would have developed it. Now we're developing it now. So you just want to really think about these things and start developing early. And it seems like a pain because you're just waiting. Mouse models take forever. IPS, everything just takes forever. But putting that investment in early on, in a couple of years, you'll see the benefits. David, I don't know if you agree with that or how you think about it.
Absolutely. I mean, it sounds like you have a wonderfully broad setup, and you're thinking about all the right things. And so, you know, we've also been very lucky that there was already a very well-organized patient group, the IFOPA, and Adam from the IFOPA was speaking here yesterday. And it's been a wonderful partnership. But no, you've hit the nail on the head there, Nasha.
Do you have anything to add, David, though, on what you consider when deciding what diseases to work on and what patient advocacy groups to work with?
Yeah, I mean, that's difficult because that's almost putting the cart before the horse. Because often if you have your own in-house laboratories where you're looking, you've already selected the diseases that you're interested in. And for us, it just so happened that there was already a patient advocacy group present, IFOPA, and again, we were very lucky that they were already very well organized, very well representing the patient population globally, ultra-rare disease, so it was very important. And like Nasha's group, they were incredibly organized and had already thought about a lot of different aspects. So we struck gold in that way. And I will just, a further plug for them is, and another thing that an advocacy group can do, and IFOPA do very well, every 2 years, they organize what they call a drug development forum. And getting together in the same room, we have patients and the families, any academic and basic research groups from around the world, any biopharma and biotech companies who are developing or are interested in developing, all get together in one room, as well as all of the treating physicians from around the world and their teams. And we all get together for 2 days, share each other's research, talk about the hot topics and how best to make the fastest progress. And right after that, they have what they call a family gathering. So families from around the world come together, and the last one was in Florida, end of last year. And there were families from Australia, South America, from Asia. And it was just wonderful to have in 1 week, everybody who's involved, both patients and the families, right up to people like us who are doing the clinical trials and our competitors. So it was a really unique experience for me. And that was all organized and was the brainchild of IFOPA, the patient group. And I think that's a wonderful initiative.
Yeah, what a huge contribution to research. So we have a question from the audience, and they're asking what advice we have for increasing patient involvement in natural history when patients may be spread out geographically. Any insights on that?
Yeah, I can tell you what we've done. We have basically almost treated it like a business where we have patient leads in different parts of the world. So we have two parents who are kind of owning the patient registry natural history study globally. And then within the US, we have 6 leads. In Europe, we have 4 leads. We have a lead in Thailand. We have a lead in Japan, a lead in China. And this is one of the things that a pharma company is going to struggle to do, right? They can't really contact parents and patients, but you as an advocacy group, you can do this. And there's so many rare disease groups where you have multiple advocacy groups. There's a lot of drama, and pharma companies hate this. And it's really, the leadership, I think, is finding a way, honestly, to move your ego to the side and find a way to work with different groups. And even if you just work together, this is an umbrella, and you build this network of patient leads around the world. And so then you've got a covered base, and then you can start recruiting patients to these studies. Our patient registry, we translated it into 6 languages. This is really critical too. You want to have multiple languages. And you can use a Google form for a registry, right? Like you can do a lot of things where translation is easy. Don't get caught up into these companies that are trying to sell you expensive registries. It's not necessary. Registries should be really cheap. It's a telephone directory with some surveys. Like it's not a big deal. So you just want to make sure there's translation, and you got to get your patients organized globally. It's like, if you do nothing else, just do that and find a way to work with people and show leadership. I think this is really critical.
And for us, for our NHS, we wanted some imaging in. So we wanted a CT scan just to be able to follow the progression of disease. And so for that, we went with a traditional CRO. So a CRO who did their work on our behalf, because it needed a protocol and ethics and regulatory approvals around the world. But working closely with the advocacy groups so that the individual country family groups, as you've mentioned, Nasha, really important. And actually, in a disease where there were fewer than 1,000 globally, we had 114 patients volunteer to be part of the natural history study. And they can often be a tough sell because there's no treatment, so there's no potential benefit. But there is the downsides of having to get on a plane often for these rare diseases, to go and visit a very specialist center, often at the other side of your country, to go through the scans and all of the assessments to keep a check and record the progression of your disease, when you know there's no potential of a benefit. And that's where I'm amazed at just the sheer altruistic perspective of a lot of these patients and their families to volunteer for these, knowing that, you know, hoping it will help them, but knowing that if not, it will help somebody, you know, in the future who receives a diagnosis for this disease. So yeah, my hat goes off to them.
Mm-hmm. Let's talk for just a minute about patient registries as related to payers. Do either of you have advice for what to have in a patient registry that would be appealing or interesting to payers that they need to see?
Do you mean insurance companies by payers, or
Yes.
just anyone?
Or, well, generally any payer, but yeah, we'll take insurance companies.
So again, I have a strong belief that, again, the advocacy group should control the registry. And if you can get someone to pay and not have control over it, that's great. But again, a patient registry does not need to be expensive. We did our entire registry around the world in less than $30,000, translated in 6 languages. And even that, I would say it should be cheaper now, because that was years ago. I would be very careful anytime you have someone coming in who's paying for your patient data to help you set it up, because you will lose control. And I just think there's nothing that's more important, because your incentives, they're sort of aligned with these different partners, right? Biopharma, academia, payers, but they're not completely aligned. And so again, and when we look at a natural history study, I just want to make it clear that there are different ways to create natural history studies. We're doing a completely digital natural history studies where patients do not need to get on a plane and go to an academic center, because 98% of the questions that are going to be asked at that academic center are found in the medical report, in the doctor's notes, etc. There's video calls you can do. You can submit like your child having seizures. There's so much you can do digitally. We're using Citizen. It's a platform that's developing these where the patients are in complete control. So I would just, entice advocacy groups to think about using technology to do these. Honestly, medical, like healthcare, is such an archaic industry that they are light years behind using technology, like the way you see other industries really embracing it. But there's a lot of things that we can do now. And I think COVID has actually helped. I speak to a lot of biopharma companies that think now, wow, a digital natural history study, that's the way to go, because we can't even get patients to come on a plane. They're looking at virtual clinical trials. So we're in the right era. And I would really recommend that advocacy groups think this way and think about control.
Thank you. Thanks, both of you. Unfortunately, we're out of time. I feel like we could go on and on, so we'll have to invite you back. And there were several more audience questions, so we'll try and get those addressed. But Nasha, for just a great conversation this morning. We really appreciate it. Before we move on to our next breakouts, I'm going to turn it over to Barbara Goodman and Claire Thibodeau of Cures Within Reach. They're going to announce the winner of our Cures Accelerator Live, and we are on the edges of our seats, Barbara and Claire, so give us the news.
Great, okay, thank you. I'm gonna just share my screen here if I can. All right. Barbara, I think you're on mute.
Unmute. Can you hear me now?
Yes.
Wonderful. Thank you so much. We are thrilled to be back here this morning. We are really excited, and on behalf of the entire Cures Within Reach team, we're really excited to present the winner from yesterday's pitch event. Cures Within Reach is a not-for-profit that finds and funds repurposing projects, taking advantage of the speed and cost effectiveness that repurposing provides. We are disease agnostic. We focus on projects that have patient impact. It's always patient first. And we really love to fund and catalyze the follow-on funding from our projects, as well as funding projects that have true immediate patient value. With that, I'm gonna turn it over to Claire, who is gonna talk about yesterday's event and the winner of yesterday's event. Claire.
Great, thank you, Barbara. First, before I do that, I'd just like to briefly thank our sponsors. We have Goldman Philanthropic Partnerships, who is a philanthropic sponsor of ours, Horizon Therapeutics, Recordati Rare Diseases, in addition to the Judy Hirsch Foundation, all helped make this event possible. And then, of course, our partners here today, Global Genes and the UPenn Orphan Drug Center, Disease Center. So for all of you who attended the event yesterday, you know that we had 5 fantastic finalists. All of them did a great job and all of them deserve to be funded. And while we only have one winner here today, Cures Within Reach will continue to raise funds to support the second place winner and beyond. So hopefully at some point we'll be able to fund all of these great finalists. But like I said, we only have one winner today, and let me tell you, it was a close vote. I know that the attendees who voted it wasn't an easy choice. They were all really great. But we do have one winner, and that winner is Angela Fleischman from the University of California, Irvine. So congratulations, Angela. Angela is with us today. I'm gonna unmute her and let her say a few words. Angela, can you hear me?
Yes, I can.
Great. Congratulations on your win.
Thank you so much. This is very exciting. I really enjoyed participating in this event and listening to all of the presentations yesterday. I know it was a really hard decision for everybody on their voting, but I just want to thank everyone that this money is going to allow me to actually do the study. Without the money, I would not be able to do the study, and so I'm excited to get moving. And make some real improvements for MPN patients.
Great. Well, thank you, Angela, and we're excited to be working with you on this this project moving forward. So again, thank you to everybody who voted yesterday. Thank you to our sponsors and to the UPenn Orphan Disease Center and Global Genes. So with that,
thank you, everyone.
Yep. With that,
thank you, Claire, and thank you, Barbara. Thank you to all of our speakers, and again, congratulations to the finalists and to the winner, Angela Fleischman. Next up, from 11:30 to 12:30 Eastern Time, you can select your next breakout session. Just go to the agenda in the auditorium, and we'll see you at our next stop. So thank you very much.
Thank you.